I'm Meredith Blackwell, and this is
Don Pfister.
and we're here to interview Reese Charvat, better known as Reese, but Iris. And she's taught here at the University of Minnesota, where we are at the Mycological Society of America meeting, and it is the year 2019. And so we'd like to talk to her about her mentors and her career and some of the friends that we had in common. So, Reese, do you?
I was so fortunate to have two wonderful mentors, mycological mentors. The first one was D.P. Rogers at Illinois. And D.P., as I have told you both, was an intellect and also a very gentle soul. And he did so many Latin descriptions or translations for people in this society of their abstracts when they published in Mycology, but also in so many mycological journals. I mean, worldwide, he would do this for people.
Me too, because I couldn't do Latin. Dr. Alex recommended him.
Yes, and of course, Dr. Alex and D.P. Rogers were friends,
Yes.
both working on
Well,
different organisms.
Right, but they were good friends.
Yes, I think they might have both crossed paths in the Iowa lab.
Yes, they did
Yes.
with Martin.
Yes, with Martin. I think they both got their degrees with Martin.
Dr. Alex didn't, but he worked with, that was
Yeah,
one of his early
yeah.
jobs, and he worked with
Well,
Martin.
D.P. did.
Yeah.
He got his degree with Martin. And it was D.P. who encouraged me to, first of all, he told me about NSF. He told me about NSF, but the one I want to talk about today is the Mycological Society of America. And he said, you really should go to these national meetings in Colorado. And there he introduced me to so many mycologists. I didn't realize the depth of this area. I meant there were medical mycologists there, people studying interactions between insects and fungi, just like here, but this is far more advanced at these meetings. But you asked at some point when we were talking earlier about Henry Aldridge and meeting Henry. I met Henry at those meetings, and we were, I wasn't then working on slime molds, but Henry was.
Right.
And we got to run around the mountains of Colorado and look and collect all sorts of fungi, most of which I did not know except for the limited amount I had learned at that point. But it was such an enjoyable time.
And you were both doing your electron microscopy.
Not yet.
Oh,
Henry,
you weren't yet?
Henry, I hadn't even decided on a PhD. It was like I said to some of you, click. Right at those meetings, I thought maybe I should apply for a PhD in some program.
Yeah.
And so that's when I went to the University of California at Santa Barbara and met Ross, Ian Ross. And he had recently come from Yale, as so many of that young faculty. They had sort of cleaned out Yale to, increase the population and the growth of this university.
Well, it's a beautiful place. So.
Yes, it is. And it was beautiful then. And so Ian was my second mentor, and he couldn't have been more different from D.P. Rogers.
And was he called D.P.? Or do you just call him
I
that?
call him D.P. I don't even know. Don is what most people.
Donald.
Yeah.
Donald. But
Rogers,
we shortened
But you
it
call
in
it
our.
Okay, that's cute. I was curious.
I should say Dr.
No,
Rogers,
DP is
because
good.
for a long time we called him that until we, got our PhDs.
I always called Dr. Alex Dr. Alex.
Well, I also worked after I did some work with Catherine Esau, and we always called Catherine Esau Dr. Esau. But anyway, things have changed, and I was going to say a little bit more about Henry, that he was incredibly excited about all fungi.
Yeah.
I didn't quite realize the depth of his research with slime molds at the time, because we were just doing field collections. But it was such an enjoyable time, and at Santa Barbara, Ian just simply opened up a new bag of tricks.
What was your thesis?
It was on plasmodial slime molds. It was electron microscopy. And of course, that's why I later went to work with Catherine Esau, because I did, as Henry did also, localizations, enzyme localizations, and it was such a primitive time. You know, I meant such wonderful markers now available.
Well,
that this is just,
But they were very good markers.
well,
the ones
you know,
that
you put a little brick in the structure, and then it improves and changes. But Henry and I both were extremely interested in these organisms and their structure function. It was a heyday of cell biology.
Yeah.
And you know, now it's advanced to being molecular, which is great, because it's far more advanced than the questions we were able to
But you know,
ask.
still, I think today there's really a shortage of electron microscopists or microscopists
Yes.
of all sorts. And people are always looking for someone to do
Yes.
some sectioning and
Yes.
and looking at something, and it's just another group of markers that you
Yes.
can use.
And I'm sure people looking at those publications would say, why on earth were they so impressed with sectioning of spores and stuff like that? But we had to make our own glass knives. You know, we didn't and we didn't have these diamond knives.
You had to go find old glass, old glass.
But
and
you
could
had
always
to
tell it 'cause
break
was-
it.
Yeah, yeah.
Yeah, I mean, this alone should have given us a degree. But the thing I wanted to say about Ian's course was that it was different from Don's course. It was extremely, very experimental in that the same thing, well, the ideas came, I think, from Ralph Emerson, just like Tom Bruns explained at these meetings about the origin of certain parts of John Taylor's course, now taught at Berkeley, that the originator of culturing different stages of so many different fungi and having them available to students to study was so different from what I saw in Don's lab. But in turn, in Ian's lab, there wasn't this huge array of mushrooms, you know, collected and studied. But Ian had put a camera, what is it, Polaroid camera on top
wow.
of a good microscope, one of his own. And then the students would use their student microscope to look at or to find the different developmental stages. And he would have them take their own photographs and incorporate them into their lab manuals. So I thought, wow, this is a way to really get students interested not only in the organisms themselves, but really making a permanent collection of those organisms for at least some time. You know, ultimately
It must have taken a lot of film.
it did. And remember, California was loaded with money then.
Oh, okay.
I'm sure this wouldn't happen now, but just that idea. And I also remember I was his TA in this course when I first arrived, and must have been less than 3 days when he handed me sheets of what I needed to do in order to address all the needs of growing cultures for the course. And it began by saying, you need to, he wasn't explaining this to me, it was on the sheet. Go to Hope Ranch, get, horse dung, make dung cultures, and that was just the start of the assignment.
Yeah, you had your whole semester plotted
Yes.
out because of when the cultures needed to start and
Yes,
so forth.
and all of the different types of cultures, potato, what was it? Potato dextrose,
Yeah.
auger. But there were so many, and it was just simply eye-opening.
Is that how you taught?
I didn't get the chance to teach a mycology course. I didn't know that at Minnesota, mycology was taught in Plant Path.
Oh.
And I was in the botany department, and I was so pleased to be in the botany department. I mean that. I should go back and say both of my mentors, Ian Ross and Don Rogers, were such helpful, generous people, and certainly helped me along the way and helped me get a position at Minnesota. But in the botany department, Dave McLaughlin had very cleverly set up a course of lower organisms, the fungi and the algae. So I was recruited, to teach in beginning courses, not just that, but that's where they needed help. And so I started in general botany, but you can incorporate a lot into a beginning course.
Sure.
And so that began my crusade for everyone knowing something about some of these very interesting organisms, including plasmodial slime molds.
I remember I didn't know what a plasmodial slime mold was when I was in grad school. I was in a PhD program at Texas in zoology.
Really?
And I was a TA, and I thought that the oatmeal on the plate was the slime mold. That's how bad it was. That was the year before I started working for Dr. Alex.
How did you change from, why and how did you change?
Well, I didn't like my advisor. I didn't get along with him very well.
Right.
And he was a very famous ichthyologist. And so I quit and looked for a job. And the only job I could find was with Dr. Alex. And we were both leery of each other, but it worked out fine in the end.
And you joined his lab as a graduate student?
Well, I worked for him first, and it was because of Henry. Henry Aldridge got a fellowship, and he had been on a TA with Dr. Alex, isolating airborne slime moths.
Yes.
And so when Henry took this fellowship at the last minute, he left this position vacant. And here I was, a zoologist, but I had had a lower plants physiology course because I had to have a botany course at Alabama.
Yes.
So
that is so interesting.
But it was because of Henry. So we both
Yes.
had things happen because of Henry.
Yes. And of course, Henry and I were in contact for many years.
I know.
When we were both working on plasmodial slime molds.
Something interesting happened about the month before Henry died of his kidney problem. The cover of Mycology had a big lizard on it with fruiting bodies of the slime mold. And some people had called him in to work on this project. They had this lizard they had captured in the tropics somewhere.
Yes.
And apparently a plasmodium crawled up on it and fruited. And so this was an article, and they picked this picture, this photograph that I'm sure Henry must have taken for the cover.
Sure. I bet he did.
And that was the month he died. So it was bittersweet.
But also he contributed so much to this society early on, and was the president of this society.
Yeah.
And again, changes are occurring now that will be very beneficial to increasing the efficiency of how our Constitution works. But Henry was always doing things that facilitated.
And especially he helped with IMC2.
Yes.
Yeah,
Yes.
get that all planned.
Yes.
Yeah, and by by the time, though, that he died, a lot of people didn't know, although he was relatively young. A lot of people didn't know him because he had started working on bacteria, I think.
Yeah, well, we all fade pretty fast.
Yeah, it doesn't take long.
It doesn't. You know, I look at this group and there are so few people that, well, so many people, I should say, aren't here that once were here and extremely active, including so many of our presidents, you know.
Yeah.
Well, it's one of the reasons to do a project like this that Meredith has instigated.
Yes.
To let people talk about D.P. Rogers. You know,
Yes.
Who would know?
Yes.
And, you know, I worked with him primarily when for IMC 2 in Tampa. He wrote a history of mycology in North America. And that somehow, or some way, that came to me as the responsibility. He did an augmented edition or something.
There was a second.
Yeah, and so I was helping Emery Simmons, I think. And Emery said, well, why don't you put together this part of it? We had the manuscript plus the augmented version. And I designed a cover, and the cover has all those signatures on it. And so he and I were back and forth a fair amount about getting that into print and so forth. And you know, he objected, as you would know, he objected to the way in which words were broken at the end of the line.
Yes.
Oh, you can't break that word here. You know, we just
There's
went
a
through
rule.
It's a rule, and you can't do it that way. And we went through the whole thing, and he was very particular about how this was supposed to be done.
As was Emery.
As was Emery. As was Emery. Yeah.
But DP served as a mentor to so many undergraduates as well as graduate students.
That I don't think we know so much.
Yeah.
Well, he turned on the undergraduates to mycology because he taught also some general courses, and he had a way of, well, the undergraduates also took his mycology course. That was the primary way. And in fact, Tom Bruns is a fantastic example of being introduced to mycology through D.P. Rogers.
Is that right?
And as I had lunch with Tom, but even before that, I was saying Tom and I are the only ones I know left in this society where D.P. was our original person who turned us on to fungi. And of course, Tom went on to Dave McLaughlin's lab, and Dave also went down to D.P.'s lab to look at collections in order to write a grant proposal, I believe. But he definitely went to see Don's collection.
At Illinois.
Yeah, at Illinois, and he was always so generous with people, helping them. And his rare book collection, if you were a graduate student, you got to use that collection. Not as I don't think as
No,
an
probably not. I can imagine.
That was a fantastic collection.
Yeah.
And I was so sorry. His wife thought it would be better if they moved to Washington State because their daughter was there, and permanently. And it turned out not to be the best thing for Don because he was separated from all of his connections. And unfortunately, Illinois did not end up with that wonderful collection, even though it was where he spent his entire academic career, I believe.
He was at New York, at the New York Botanical Garden.
That's right.
Before he went to Illinois. Yeah.
Yes, but his main part of his career was spent.
Yeah, and he was in New England for a period at, what was it, American University, in someplace there. But he used to come to the Farlow in those early days, not when I was there, but earlier. And he did a lot of work in the collections and did translations and other things. And
Yes.
So really quite a wonderful array of activities for
Oh,
him.
yes.
So did he have a special facility with languages, or was it just Latin?
I only knew about the Latin because we saw that happening,
Yeah.
and it was frequent. What was else? There was some other thought I had about Don that it's gone. Maybe it'll come back. But anyway, he was just simply a wonderful person.
Yeah. Well, he did this early work on the basidium and the development of the basidium, and then worked on Corticiaceae and some of these other crust basidiomycetes and so forth. So he did a lot of systematic work.
Well, I remember now what I wanted to say because you spoke about some of these organisms. Someone in one of the symposia mentioned splitters and lumpers, and they were looking. It might have been Joey.
Vetiver?
Yes, I think it was Joey, and talking about, even with this complex, these wonderful, powerful tools that permit people to put organisms in groups where obviously they're more closely related than when we were doing it 20 years ago. But of course, DP and Donk in Europe.
Oh. They were a little bit like this.
But they were splitters and lumpers, so I was so amused as soon as I heard that phrase, thinking, DP and his addressing of systematic problems. It was quite.
Was he a splitter or a lumper?
Well, frankly, I never could quite figure out, depending, I think, on the
organism.
The organisms.
Well, Dunk created lots of genera.
So at least
divided things up probably more than Don Rogers
would have permitted.
Yeah.
Yes.
Yeah.
But anyway.
So you're still living in Minneapolis then?
Right, and if you want me to, I'll say a little bit about my own graduate students.
Yes, that would be good.
Again, when graduate students come to your lab, they come with different needs and intellectual backgrounds and different wants. And it has always amused me how on earth we end up with the wonderful graduate students that come to our labs. But I know when Teresa Pawlowska was checking out labs in the USA, she'd come from Poland. And the first thing she said to me about in terms of academic topics was, well, I need a heavy metal site. And all I can say is, thank God for Pig's Eye, a Superfund site nearby, because we got permission from, the Minnesota EPA to go on that site, and
so she knew what she wanted to do.
Where she had worked with heavy metals in Poland, and so she had really a wonderful background, and I'd always hoped she would go back to them, but when she went off to a postdoc in John Taylor's lab, and then I heard all of her talk today, which I hadn't heard her talk in the last couple of years, I thought, I might as well get rid of every last sample of heavy metal soil.
Maybe over.
It is over. But I was delighted with all of the work that she's done with so many different groups and with John. It's just simply, wonderful.
She's done really well, and she has a very good postdoc now, Chase Myers.
I just heard him talk. It was a wonderful talk, yes. And the other thing, I was going to talk a little bit about Pete Avis, who was here, but they just had a baby, this year, and so Pete he said, I didn't give a paper, and I thought, thank God. I meant, you need to take a little break now and then, and both Aaron and him need some time to recoup. And when he came to my lab, he was very interested in AM fungi and looking at AM fungi and incorporating into their development and whatever he was going to do all of the knowledge that he had learned in the ecology courses he had taken it as a master's student. But it soon became evident that this wasn't going to happen because we had no good molecular probes at the time. So he hooked up with Dave McLaughlin's lab, and Dave and I both became a mentor, as well as Patrice Morrow, head of head of ecology, and also just a really nifty ecologist. So he put together his own package of mentors, and everything worked out because he, and also Ian Dickey, I don't know if you met Ian.
I haven't.
He also works on fungi, but now in New Zealand. He was a postdoc in the forestry lab, but it was a group working together on the ectos, and they did all their dirty work in my lab and all the clean work in Dave McLaughlin's lab, which was right down the hall. So it worked out well.
Yeah. So how did you get to AM fungi?
Oh, it's my students.
Your students.
I came to Minnesota thinking I was going to continue working on slime molds. But Carol Deppy from Harvard was there already, and she was working on schizophyllum and talked to me about these neat mutants that she had. Not her, it came out of Raper's lab,
John
Red
Raper.
Raper's
John
lab.
Raper,
Yeah, and my God, they were morphological mutants where they knew so much about the genetics. And so we were going to team up, and I was going to work on the cell biology, and she already was well into the genetics and also some of the cell biology. But in the end, she decided this wasn't the career for her, and she went to the West Coast and set up a more practical setup.
Okay.
Oh, and then what was I going to work on? Well, I wasn't particularly, I was interested in AM. It's just that it was really Charles Vaubel that none of you met. He was a master's student who again took a Plan B, but he was very interested in AM in wetlands. And so he collected cattails and was looking for AM fungi in cattails. And a lot of people, particularly early on in the 80s and early 90s, I think we were interested in if emergent aquatic plants had AM fungi, and if they played any role or were just parasitic or whatever. We wouldn't, anyway, were they even present, and what structures were present? And that's how we started working on it, and so many students were attracted to the lab, and undergrads wanted to work on the organism. And one of my fantastic undergrads, when I say mine, it was only because Jen White decided to do an honors in my lab, and she worked on the relationship between arbuscular mycorrhizae and phosphate concentration and the roots of purple loosestrife, a classical sort of experiment that many people were doing at the time. And she got very neat results that were published in a mycological journal. That, of course, it's a facultative mycorrhizal fungal plant, and it kicks out the, MA fungus when high phosphate is present. But now this is well known for so many of those plants.
Right.
Yeah. But also the point is that a group of these experiments were done in my lab, and then the state had money available to look for these organisms statewide, and some of the some of the studies we'd done weren't what I would describe as heavy-duty scientific studies, because when you work with the state agencies, they need certain information, and it's not information necessarily that you might want to go out and get, but we did. And we learned an awfully lot about plants and distribution of AM fungi throughout so much of Minnesota.
Yeah. Well, Minnesota is very good about natural resources and
Yes.
funding
Yes.
surveys and other work.
Yes, they
so,
want surveys done.
Yeah.
They want to know this material.
That's great.
But a whole group of students then came in to do restoration with AM. And Dan Tix, one of them, you wouldn't know Dan because you know, he is a person who's actually doing the restorations. And now it's primarily wetland restoration, but he started with prairie restorations and studying EM and plant distribution. And the truth is, it seems like every restoration done on a slightly different soil type has different requirements and different fungi being present, or if you add them, actually it does improve a restoration if it's on soil that lacked them, and if it's along highways where we have worked, and that was Smith, Michael Smith's masters that proved to be very interesting and showed that there really was an advantage of adding these AM fungi, but of course they needed to be produced from a natural site within a 25-mile area, or at least a limited area. The idea of spreading them all over from many different sources, probably ecologically is a bad thing, I would say.
Yeah. And they're probably slightly endemic, at least.
Yes.
Yeah.
Yeah. And I don't have information about it. It's just we tried to be conservative and follow ecological models for seeds, because frankly I think sometimes we muck up everything by not knowing enough and trying so hard to do things.
Well, this has been great, I think, to hear from you about your mentors, about the projects and so forth. Thank you.
Thank you very much. Thank you for giving me the opportunity.